wasting much of it. See, I will start from paper 2 again. Now, my screen question number 1 is visible to all.
Yes, ma'am.
Okay. Now, question number 1 will have 3 options: enumerate the erythrocyte, enumerate the leukocyte, or differential count. So, if the enumeration of total count of erythrocyte or leukocyte is asked, so dilution and charging will have 4 marks, counting in 1 microsquare 4 marks, calculation and result 4 marks, 3 marks are for the explanation. Now, second slide is visible to all.
No,
Yes?
ma'am.
Not visible.
No, ma'am.
Just a minute. Huh? No one will on their video, please. This is a humble request. Now, second question is visible to all.
No, ma'am.
Still no.
No, ma'am.
Alarm, I request to switch off your camera. What is the problem? Now?
No.
Now? Still no.
No, ma'am.
Still no. Just
Yes, ma'am. It's not visible.
okay. Just a minute. Huh? Now? Question
No,
number 2
ma'am.
is
No. No, ma'am.
just a minute. Let me check what is the problem. Now?
No. No, ma'am.
Okay. Now I think there is a problem. It's not visible, na question 2.
No, ma'am.
I think initially it is happening that it is not getting visible and later on it's visible. Now,
Yes, ma'am.
okay, now you all can see the question. Let us discuss the question number 2. Now in question number 2, I think question number 1, differential count was left now. Now differential count is there on my screen.
No, ma'am. No, ma'am.
Yes, ma'am. Now it's visible, ma'am. Second question.
I'm asking for a differential count question.
Yes, ma'am. Now
Yes,
it's
ma'am. Visible.
All right. So for differential count, 3 marks for preparation of the smear. That means you will be taking a drop of blood and spreading it on a slide. That is called as the preparation of the smear. So for that you will have 3 marks, staining 4 marks, identification of at least 2 type of cell 4 marks, and its a result and explanation. So basically they will ask the viva on it. Question number 2 is visible. Question 2 is visible now.
Yes, ma'am.
Question 2 only now.
Yes, ma'am.
All right.
Yes, ma'am.
Okay. So for question number 2, you basically should know the chemical reagent principle, reaction mechanism, and for that you will be getting 15 minutes to write and 3 marks. Okay. Basically, initially the examiner may ask you to write the aim requirement principle. So they will check it, and after that only they will allow you to perform the practical. Now, for doing the experimentation, observation table formula calculation part, you will be having 5 marks. Knowing the normal values of protein and triglyceride, you have 5 marks, sorry, 2 marks, as well as you must know the clinical significance and diagnosis of it. Diagnosis means which disease is there. If this particular situation is there, then what is the, now how to perform the total plasma protein. So for performing that, you should label 3 test tubes as standard, unknown, and blank. And in first standard test tube, you will be pouring 1 ml of BSc standard solution, 5 ml of alkaline reagent. For unknown, you will be adding the serum sample and 5 ml of alkaline reagent. Then in blank, you will be adding the 1 ml of distilled water and 5 ml of alkaline reagent. Now see children, the purpose of making blank in any assay is just to calibrating the machine with the chemicals color. Okay, this is our basic aim. Otherwise, you will not observe that any roll of blank is there. Okay. Now, after preparing these 3 test tubes, you will be waiting for 15 minutes so that the reagent will react thoroughly with the alkaline BSA or whatever your serum. And after that, you will be adding the pollen COKL to reagent 0.5 ml. After that, you should keep it into the dark for 30 minutes and you will be taking the OD. Now next. Now on my screen, plasma triglyceride phosphovanillin reagent procedure is visible.
Yes, ma'am.
Yes, ma'am.
Okay.
Yeah,
So
ma'am.
in that, step 1 is the digestion, step 2 is the estimation. Now digestion is what? You will be taking the serum or whatever, you will be digesting it with the sulfuric acid. So one in blank, you will be adding 1 ml of distilled water and 2 ml of concentrated sulfuric acid. Be very careful while using concentrated sulfuric acid. And in standard, you will be taking 1 ml of standard lipid and 2 ml of concentrated sulfuric acid. In sample, you will be taking 1 ml of serum or whatever plasma is there and 2 ml of concentrated sulfuric acid. Now, thoroughly mix this content. Be very careful while using it and keep the tubes into the boiling water bath for 10 minutes. Now, say student, whenever you are keeping the test tubes into the boiling water bath, make sure that, who's this? Please, Alisha Khan, switch off your video and audio. Mix the content, sorry, whenever you're keeping the things, say student, when you unmute, you suddenly start sharing your video, it disturbs the meeting. So please maintain the decorum and continue. If you are not comfortable, you are allowed to leave the meeting. All right. So whenever you are keeping the test tubes into the boiling water bath, you should be very careful. You will be just plugging it with the cotton. Now I have already taught you how to make the cotton plug, and if you don't know, it is there, recorded video is also there, you can see it. But make sure before keeping it into the boiling water bath, you will be plugging it with the cotton carefully. Then after that, you will be using this particular mixture for the estimation. Now step 2 पे आ जाते हैं. Again you will be preparing.
बार-बार डिस्कनेक्ट हो रहा है.
Just a minute. From my end, there is no such problem. I have checked the signal strength. Okay, let us continue. Now in the estimation step, you will be taking the blank में blank वाला जो test tube बनाया था 1 ml distilled water and concentrated sulfuric acid, you will be utilizing same. Now see, while taking mixture from step 1, be very careful that contains sulfuric acid. No one is going to pipette it from the mouth. We have pipette pump in our college, so you will be utilizing the pipette pump for that. All right. I'm extremely sorry for that. Now in that, you will be adding 3 ml of phosphovanillin reagent. बच्चों, phosphovanillin reagent also contains concentrated orthophosphoric acid in that. So here again you should be very careful. Standard same standard वाला जो digestion है, that mixture you will be taking plus 3 ml of phosphovanillin. Sample भी में sample वाला mixture लेंगे, then you will be adding 3 ml of phosphoenolin. Now you have done with 3 test tube preparation. Now you will have to imagine the regular practical. Those who were there, they will recall it better. After that, you will be keeping it at the room temperature for 15 minutes. A pink color will develop. You have to take the OD at the 540 nanometer. Now question number 3 पे आ जाते हैं. On my screen, question number 3 is visible to all.
Yes,
Yes.
ma'am.
Now question 3 में भी बच्चों we have 3 options. Okay, in that I'm going to explain 2 only. Estimation of hemoglobin and LDH. So we have first option, estimation of hemoglobin by salicylic method, ESR and serum LDH. Now for first and third option, for knowing the chemical reagent principle and reaction mechanism, you have 3 marks: experimentation, observation table, formula calculation, 5 marks, normal value and significance, as well as diagnosis. Means अगर ये particular चीज present है, so what it diagnose? It is diagnosis of what? Disease. So that you should know for the 2 marks. Now estimation of hemoglobin by salicylic hematite method, in that you will be filling the graduated tube. बच्चों graduated tube में 2 markings है, एक red से है, एक yellow से है. That's why I have stuck this photo. Okay. तो जो red वाला marking है, in that you will be filling n by 10 HCl up to the 20% mark. Now see here, be very careful while doing it. Majority of the student will be using the yellow marking. इसीलिए मैंने ये डाला intentionally. So you fill the graduated tube up to 20% mark. Now HCl, n by 10 HCl is given to you in your kit only. Okay. Now what are the components of kit? I will show you first. So you will have these things in your kit, but this thing will not be there. Okay, this pipette will not be there. Instead of that, we will use the micro pipette. Now this is your comparator box. This is your graduated tube. This is a stirrer, and this is a dropper which you will be using to add the drop by drop this water. Okay. Now let us come back to the procedure. Now first you will be filling the graduated tube up to the 20% mark in red with the n by 10 HCl. After that you will be filling your micropipette with 20 microliter blood, or the same you can use the pipette given in your kit as well. After that you will be pouring the blood into the pipette which is containing the n by 10 HCl. Now allow to stand for 10 minutes so that the HCl will react with the hemoglobin. Now see, after the 10 minutes only, you will be start adding the distilled water in that. Now, till when you have to add the distilled water? Till the brown color of HB tube, this graduated tube, matches that of standard solution comparator glass. This is the comparator glass. यहाँ पे हम लोग हमारा graduated tube रखने वाले हैं. So this 3 colors should match. तो उसको थोड़ा सा ऊपर लाइट में देखेंगे. If it is perfectly matching, तो आपको graduated tube वाला apparatus वाला slide दिख रहा है? Yes, can you see?
Yes, ma'am.
All right. So you have to compare it. If it is comparing properly immediately, you will be showing the result to the examiner. Now determination of serum LDH, ये तो simplest है, इसमें कुछ ज़्यादा है नहीं. Only how to take absorbance into the calorimeter and spectrophotometer. I have already explained you. I have already posted the video of it. लेकिन calorimeter का एक बार फिर से I'm going to show you video with the instructions. Okay. Now determination of serum LDH. So in that serum sample you will be taking 30 microliter. Now see, micropipette will be there. Please note it. You will be taking it 30 microliter only. Okay, there is a vast difference between the microliter and milliliter, huh? So don't do this thing. Don't waste the chemical there. 30 microliter serum sample you will be taking. After that, you will be adding 1 microliter. 1 microliter is equals to 1 ml working reagent in that. And after that, you will be waiting for 1 minute. Wait for the 1 minute stopwatch will be there. Then after that, you will be taking your first reading and you will be taking the reading 4 times. Okay. Now see, first reading you will be not counting. Baki ke jo 4 unit hai na, which you have taken on 340 nanometer, you will be taking the mean of that, and that is going to be your absorbance formula. All right. Is that clear? Now how to take the reading?
Yes, ma'am.
Just a minute. Huh? Let me show you how to take the readings. चला गया. My screen is visible. Is my screen
Yes,
visible?
ma'am.
All right. Okay. Can you see this folder?
Yes, ma'am.
Okay. Can you see the video now? Yes or no?
No, ma'am.
So first of all, just a minute. It's not opening. Just a minute. There is some problem. Just a minute. There is some problem. Stop share. I think I have already posted it on YouTube. I will share that link with you. Okay. Now, this web page is visible to you all. Yes or no, बच्चों?
No, ma'am.
No, ma'am.
No, ma'am.
Okay, तो बोलो ना no. Just a minute. Just a minute, बच्चों. You have to go very fatafat because of the time limit. Now this is visible. Yes or no?
Yes.
Yes, ma'am.
Okay. Now let me stop. Now see, this is our colorimeter machine. So when we will be using this colorimeter machine, so now let me remove my hand, then I will explain. I will explain you with the arrow. Arrow is visible, na student? Arrow is
Yes,
visible.
ma'am.
Yes, ma'am.
Alright. Now see, this machine have here the transmission. जहाँ पर मेरा finger नजर आ रहा है वहाँ पर absorbance का button है, ठीक है? यहाँ पर next button जहाँ पर मेरा arrow है is the unlock button. This is the calibration button. This is the slot where we are going to keep the most important care while using the this colorimeter is you should not keep this slot empty, okay, because this is the slot from where the light is passing. When you keep the slot empty, so thoda bahut jo hai, there will be a problem into the machine calibration. So first we will calibrate the machine with the distilled water. Now this is the transmission mode. First of all, you are going to unlock. When you will unlock it, now can you see here this light? My arrow is visible not to all. Yes or no? This arrow is visible to all.
Yes, ma'am.
Okay, very good.
Yes,
So
ma'am.
when you will press this unlock button, one light will blink here into the unlocked side. Okay, so now your machine is unlocked. You can calibrate it. Now here this T% is written. That is your transmission button. We'll press transmission, and here the blue button of calibration you will press after that. So here you will see the 100% transmission. Now here where my finger is there, there is an absorbance button, and absorbance button ABS is written. So after that you will be pressing the absorbance button. Okay, once you will press the absorbance button, so here the light will blink into the absorbance slot, and again you will calibrate it. So once your machine is 000, 0.00 is it is calibrated. Okay, now you are going to fill the another cuvette. All right, another cuvette may you will be taking your blank. Why you will be taking blank can be asked as a viva. So in that case you should be able to answer if we do not require the chemical colors, we just want to observe the chemical reaction happened with either the plasma or the standard. That's why you will not take the colors of ब्लैंक. तो जो केमिकल के कलर्स को एक्सक्लूड करने के लिए, जैसे कि बच्चों व्हेन वी विल बी टेकिंग द weight into the weighing balance, तो whenever whatever petri dish weight is there, that is not required. So what we do, we use the tare option ना, तो यहाँ पर हम लोग उसको blank से calibrate करते हैं. Okay, we will be calibrating it blank. I have already explained you the procedure of blank. You will be unlocking your machine, you will be pressing the transmission, after that you will be pressing the calibration, and you will be getting 100 into the digital window, and after that you will be pressing the absorbance and again calibration button, and after that you will be closing the lock option, यानी कि उस बटन को फिर से प्रेस करना है, so your machine will be locked and you will be taking the reading of standard as well as your unknown. Is that clear? किसी को कुछ doubt है how to use the colorimeter?
यस मैम, वो बीच में आपको समझा नहीं था अनलॉक करने के बाद.
नहीं समझा था. I will
मैम बार बार है ना डिस्कनेक्ट हो जा रहा है हमारा.
Okay, but बच्चा I am recording it, so you will be getting the recording of it. Okay, बेटा?
ओके मैम.
Don't worry about it. But
Okay, ma'am.
Kalam
Okay.
has asked me the doubt, so it's my responsibility to clear it. Bacha, when you will unlock the machine, so there are two options. One is the transmission, another is the absorbance. So first you will be pressing the transmission T% button, and after that you will be pressing the calibrate button. Once you have pressed the calibrate button, you will get 100 on your digital screen. Already this video is shared with you. So again, I'm explaining from the same video only because right now I don't have any colorimeter. So next, you will be pressing the absorbance button. There you should get 0.00, and after that, you will be again locking the machine. And once you are done with taking your blank reading, that means your machine is calibrated. Now you will be calibrating machine with your blank. Now we know the purpose of blank, that I had already explained you, that you don't require the colors of chemical, you just require want to know the reaction color. So for that you will be taking the blanks calibration. So blank के ऊपर machine को फिर से calibrate करना है, and after that you will be taking the reading of standard as well as your lipid. Is that clear? Shall we move ahead?
यस मैम.
We cannot stick to it. As I told you, there is a time limit of it. All right. So let us move on to our PowerPoint presentation. Now
Okay,
can
ma'am.
you लेटेक्स अग्लूटिनेशन टेस्ट के ऊपर आ जाते हैं. सीरम एलडीएच में किसी को कोई डाउट है? Any doubt?
Ma'am, only colorimeter will come or we will have to do for spectrophotometer also?
It depends on the exam. The spectrophotometer, I have already shared the videos. Same way you will be taking reading into the spectrophotometer. Only the thing is that you have to I adjust the absorbance, and you will be taking care of spectrophotometer. Now that is already shared. I cannot take that also because of the time limit. Okay, now determination of serum LDH is visible on my screen. Yes or
Yes,
no?
ma'am.
Now question number 4. Now question number 4. Now can you see the question number 4?
Ma'am, what about Western green?
Can you see the question number 4 now?
No, ma'am.
No, ma'am.
No, ma'am.
Just a minute, just a minute. Now?
Yes, ma'am.
Yes, ma'am.
Okay. Now question number 4. Perform the latex agglutination test, rheumatoid arthritis, 5 marks, or record bleeding or clotting time, and comment on the clinical significance. For both procedure and setup, 2 marks. Result and conclusion, 2 marks. Explanation, and now here in the explanation you should be able to answer the clinical significance of it. So latex agglutination test. Now see, there is a kit, commercial kit we are going to use for it. In that there is a dropper. You will be, by using the dropper, now here you will be getting the blood. Okay, so you the blood you will be pouring into this black slide will be given to you. In the black slide, in one circle, you will be putting 1 drop of this blood, in next negative, in next positive control. So all this thing will be there into the kit only. Only serum will be provided separately. Okay, bacho. So after that, there will be a small stirrer will be there, white color. So with that, you will be just mixing that latex reagent in that. After that, you will be thoroughly, after mixing, you will be observe the agglutination. Now see, in the negative you will not get any agglutination. In positive you will get the agglutination. All right. Now next practical, bleeding time and clotting time. For bleeding time and clotting time, you will be sterilizing your finger tip with the alcohol, 70% alcohol. Now dry, prick the fingertip with the sterile lancet. Start timer and note the time of bleeding. After every 30 seconds, blot the drop on the blotting paper and note the time. Now see, you should be very much careful that you will be not touching your finger to the blotting paper. After that, stop the timer when the bleeding stops. Note down the time. In bleeding time over clotting time, sterilize the fingertip with the 70% alcohol. Allow it to draw. Prick the fingertip with the sterile lancet. Collect the blood into the capillary tube. Now see, when you will be collecting it into the capillary tube, you should take care that you should not hold it very slanting. You should draw, hold it very properly so that the blood can be pulled into the capillary tube. After that, start the timer, note down the time after first 2 minutes. Now after 2 minutes, you will be just keeping it like this. After the 2 minutes, you will break off a small piece of capillary tube, check for fibrin thread. Now see, checking fibrin thread is very, very important in this particular practical, because that a very small thread will form. Otherwise, you will keep on breaking the capillary, you will not get that fibrin thread. Okay, so once you feel कि blood थोड़ा clot होना शुरू हुआ है, that time you will be noting as a clotting time. If the fibrin thread is not observed, so a small piece after every 30 seconds you will be breaking. Now next is Viva Voce, which is based on the theory for the 5 marks. Your certified journal is for the 5 marks. Any doubt? Any doubt? Yes. Is
No, ma'am.
anyone has any doubt?
मैम क्वेश्चन नंबर 3 में विंट्रोप मेथड है वो.
Okay, I will explain the Winthrop's method. Now, see, Winthrop method basically, it is a very simplest thing. You will be having one Winthrop's tube. In that, you will be just pouring the blood. Winthrop's micropipette will also be there. You will be filling it till the 10 number, and after that you keep it into the Winthrop stand, and the blood will start sedimenting, and then you have to note down that difference. That's it. Okay. Anything else? Because now I have only 6 minutes 42 seconds. Come on,
Ma'am,
fast.
blood sample we will get or we have to prick our fingers?
No, you will be getting the blood sample. Any other doubt? Yes.
Ma'am, we'll get the protocol, right?
Yes.
प्रोटोकॉल्स रहेंगे